Hi, hello everyone. So today I'm going to talk about the early safety screening in drug discovery, and this is kind of a very critical importance issue and topic for all the people who are working in drug discovery. My name is Chun-Yao Lee from Eurofins Panlabs Discovery Services, Taiwan. Very nice to have this opportunity to talk with you, and we can change more opinions during this seminar. So today we are going to introduce the overview of safety pharmacology to understand and interpret what is the safety pharmacology, why it is so important, and then what kind of methodology we are going to use. Usually it's from the in vitro safety screening part, and then we can interpret the data to the in vivo and even to the clinical observations. And also I will focus a little bit more on CNS safety, cardiovascular safety, and also carcinogenicity risk assessment as some example when we are evaluating the overall safety pharmacology and the safety screening in the preclinical and discovery stage. And then we'll have some summary and some take-home message for your information. Okay, let's starting from the safety pharmacology overview, and that will be a kind of long history, and I will try to introduce these points step by step. Okay, let's starting from a very famous and very impressive example and in the beginning of 2000. So I remember when I was in my pharmacy school, and we know we are looking for better NSAID. They have a better anti-inflammatory effects and showing more selectivity in COX-2. So in 1999, FDA approved the COX-2 inhibitor called Vioxx. And this is a very popular product from Merck. But a few years later, in 2004, they just withdrew this compound from this medicine from the markets due to the side effects. Actually, they can cause kind of very critical heart and cardio attacks, and then a lot of people they just die from these side effects. So in the end, they have to withdraw this compound. And then this is the third biggest product recall in the human history. And this is a lesson of almost 9 billion. And why it happens? So there's a lot of reason, and people still taking the investigation to this phenomenon. And in the end, in 2006, Lancet researchers estimate there are 88,000 Americans had heart attacks, and also 38,000 died just from taking the Vioxx. And then in 2007, Merck announced to pay around 4.9 billion to end the thousands of lawsuits from the side effect and deaths caused by Vioxx. So this is really kind of very critical and very expensive lesson to human history. Even until 2015, FDA still strengthened such kind of warning for all the NSAIDs that can cause severe heart attacks or strokes and cause some cardiovascular side effects to the human body. So still we need to pay much attention on this kind of NSAID application. So why we have such kind of thing and why this is so important? So I think after such kind of lesson, people are starting to think about how we can try to minimize the risk and to avoid such kind of adverse effects in the very early stage and try to avoid such kind of consequence in the end. We know if we get the marketing approved and then we have to withdraw the medicine, and that will cause a lot of issue and a lot of loss of economy. So when we are talking about the cardiovascular risk, let's go back from the early in 19th centuries. Actually, this is hERG channel blocking effects, kind of our maybe very early knowledge to the human medicine side effects. So we know hERG channel is kind of a potassium channel, and they can they can maintain the membrane potential in a very stable level. But if we abstract the potassium current outflow, and that will delay the repolarization and make a QT interval prolonged. And once we have a QT interval prolonged, then we got some arrhythmia effects, and then we can suddenly get kind of a sudden death or a cardiovascular issue. and like such kind of ventricular tachycardia on ECG we can observe. So after that, we know hERG is very important ion channel, and any drugs that bind to hERG, they will have the risk of arrhythmia. So this is very early knowledge we are knowing. People we know about the side effect to cause cardiac arrest and arrhythmia. And even until now, hERG is paid by a lot of attention, and even including to the guidance, all the drugs we need to test hERG channel blocking effects. But as we know, not only hERG, but also the other receptors, channels, or transporter enzymes, they are also important to be involved into such kind of adverse effects. So here we can see the figure, like we have 4 quadrants. The x-axis is from the happening, we can say it's instance of frequency, and the y-axis is kind of human body impact. So we can see the first one, we see the QT prolongation and the ionotropic changes, especially for the cardiovascular part. And like we as mentioned in hERG. So this is very often happening, and also the impact is very high. Even we have some research rates from such kind of adverse effects. So they are in the first quadrants. But also we need to be careful and pay attention to the second quadrants, like neuropathy, like seizure liability, abuse liability, bronchial constriction, or respiratory depression, such kind of side effects. Even though their instance is relatively lower, but still they will cause very high impact to human body and health. So still we need to pay attention on such kind of adverse effects. And this first and second quadrant are really be paid a lot of attention when we are doing the assessment of safety pharmacology. But also there is also some other attention from the third and fourth quadrants, like motor dysfunction, cognitive dysfunction. Maybe their incidence is very low and impact is low. And also such like very frequently we see the heart rate change or postural hypertension, some like change in blood pressure, or some very commonly observed like sedation and GI dysfunction. Although they don't cause a lot of very huge impact to human body, but still that's cause some like uncomfortable feeling, and also to affect the compliance of human when they are taking the compounds, the medicine. So still, if we want to do a better, good medicine, still we need to pay attention to such kind of relatively minor but still frequently happened to influence patients' compliance compounds. So that is also something very important we have to know. But of course, when we are doing the critical safety assessment, we focus on the first and second quadrant, like QT prolongation, arrhythmia, drug abuse, seizure liability, such kind of high impact phenomena. So I think we already know pretty well regarding the ICH guidance, talking about the safety pharmacology. And actually this is initiated from the 2001. And in the guidance, we talk about the tier 1 and core battery system. They are mostly responsible for the central nerve system, cardiovascular system, respiratory system. They are directly involved and impact the human life and function. Like we don't want to have any adverse effect on the brain, on our heart, or our lung. So this is kind of a core battery system, and they are quite important listing the tier 1. And also we just mentioned they are kind of a minor or low impact side effects, like a GI dysfunction, like renal system. They are listing to the tier 2, like supplementary system. Although they don't have an immediate lethal problem, but still for chronic usage, they still need to be paid attention, like renal system, GI system, and the others. So when we are talking about the safety pharmacology, also we need to know the basic pharmacology rationale. So what is our on targets? What is off targets? So what we call primary pharmacology and secondary pharmacology. So in this figure, we can see like when we design a medicine, a drug, or a drug's metabolites, we have our intended arriving tissue and intended target receptor or ionic channel or some protein. So this is our intended effect and intended receptor. So we call this as on target. And for this kind of medicine, maybe they are starting from chemical compounds. They will have some kind of, not only for the intended receptor, but also they will have some influence or binding on the other receptors or on the other tissues we don't intended to reach. Then we'll say this is unintended tissue or unintended receptors, and we'll say this is kind of off targets. There will be cause some physiological or pharmacodynamic effects we don't like to see. So in the right figure, we can see according to the human knowledge, this data is until 2020. Maybe we'll have a more updated information. But we know like intracellular targets, membrane targets, and secreted targets. Actually, to our human knowledge, only a very small portion we know this is druggable or this is drugged already to be a kind of a drug target. But there are more, like more than 90% or even 95% targets we don't know. It's in human body, but we don't know this is for drug targets. But there could be a lot of potential to be off target and cause some unwanted effects we don't like to see. So of course, we need to really pay attention when we are doing the on-target study and off-target study. We always need to exclude some kind of a critical off-targets to avoid such kind of adverse effect. As we mentioned, like hERG channel or like other receptor related to CNS and cardiovascular. And that could be, should be avoided. Okay. And when we are looking about the whole workflow in the drug discovery and drug development, we know that is kind of very time and cost consuming process. Yeah, before when we were like study in school and long time ago, maybe decade ago, we say this kind of a drug discovery and development takes around 10 to 15 years. And that will take around 1 to 2 billions. Even like if we have attrition, maybe that will be even more expensive, as example already we show in the Merck's case. But nowadays we have a more and more like alternative of new novel technology, maybe like in silico screening, high throughput screening, and some in vitro alternative way. And thanks to such kind of novel technology, probably we can minimize or reduce the whole process from 10 to 15 years to maybe just a few years. Some people, they claim by using the AI tool, they can minimize this whole process even within 2 or 3 years. That could be kind of very like huge advance. But still, when we are working on this early discovery and the later phase of development, still we need to pay attention about any kind of safety issue and to avoid the side effects. And we can see from early stage, the cost per compound, maybe from the like compound screening price 0.02 million, and until the lead optimization is around 1.6 million. And if they are going to the preclinical test in the development stage, that will hugely increase like to 10 million. And if we go to the clinical phase, that would be more than 100 million in the final stage, like phase 3. So that is I think that is kind of very good business if we can really pay attention at the early phase, especially on the discovery stage. We can exclude the unwanted effects or some compounds they have a high risk potential, and then we can save a lot of money in the later stage. So that is kind of smart investigation when we are doing the safety assessments in the drug discovery or preclinical studies. Okay. And then we are going to review a little bit about the history and the progress and the workflow starting from 2001 when we have the ICH guidance S7A talking about safety pharmacology. And then to 2005, we have ICH S7B and E14 talking about the QT interval prolongation. So this is like, I think after 21st century, we really pharmas and people, we are paying really more and more attention on safety pharmacology and try to create a more like safer medicine in the world. So we say this is according to our like CRO development also, we usually follow the regulation and follow the guidance. And once the guidance have some like explanation or some demands and requirements, and we try to fit. So I would say like from after 2000 to like until the paper in 2012, we say this kind of first generation for CROs, we were working quite a lot in safety screening panel. So after this 2001, already we have the idea about the tier strategy, meaning like in the lead identification to optimization to pre-clinical candidates, we have a different kind of strategy. In early phase to middle and to late phase, we'll select different kind of targets or different number of targets to do such kind of tier study. So you can see in the first generation, we start such kind of strategy. But in today's presentation, actually, we want to highlight more about the milestone paper in 2012. So both paper in Nature Review of Drug Discovery, they set the milestone of the 44 targets and the modern standard for the safety pharmacology. So regarding or related to the core battery system and some important targets, this 44 target now become kind of a gold standard when we are reviewing and assessing the safety pharmacology and the drug safety for the kind of standard workflow. So we call after 2012, this is kind of second generation for CROs. We start to follow this reputed paper and the core and important molecular targets like these 44 targets. And then we set out such kind of safety screen 44 as kind of industrial standard. And also still we adopt such kind of a tier strategy from hit to lead optimization and candidate selection. We have a tier 1, tier 2, and tier 3 with a smaller panel, less targets, to the later phase, huge panel, more targets, but small amount of compounds. So such kind of strategy will be very practical for the industry practice when we are doing the safety assessments. And also until last year, 2024, we got a new paper from the Brennan et al. Talking about the published in Nature Review Drug Discovery, talking about the modern safety assessments based on the 2012 paper, 44 targets. And in the past decades, there are more development in oncology and immuno-oncology, and also a lot of modalities working on kinase. So in 2024 paper, they mentioned about like more the importance of kinase and add some like kinase dominant supplementary targets based on the 44 targets and build up the so-called the third generation of safety screen panel. And also we make the tier strategy more like in detail, like in very early stage in hit finding. Also, maybe we just need a few targets and we call this is core panel. And then we move to the 44 and move to tier 2 like conventionally like 87 or the new paper publisher, the 77. And then still we go to the candidate selection. We have a more broad spectrum screening panel for the final candidate selection. So that will be kind of evolution of safety pharmacology to people talking about how many targets, what targets we need to select. And until with the different time trend, maybe we focus more on oncology. Maybe in the next 10 years, we'll focus on other things else. So such kind of target selection and coverage of molecular targets probably will be changing with time. But most important things like the pharmas and biopharmas and CROs, we are trying to adopt the trend and try to follow the latest progress of a drug discovery and try to propose a better and better solution for the assessment of safety and to avoid unwanted or unintended side effects by our best. So let's just look closely regarding the 2012 paper, the 44 molecular targets. So these 44 molecular targets was just kind of agreed or proposed by the 4 major pharmas 13 years ago, like AZ, GSK, Novartis, and Pfizer. So they talk about these 44 molecular targets are kind of a very core, as we mentioned in the core battery system. They are very core to our CNS, cardiovascular, and lung. Such kind of core battery system, they are kind of fundamental molecular target we need to screen no matter what kind of small molecule we are working for. And then also we have such kind of workflow from tier 1, tier 2, and tier 3. We increase the targets. And after the paper published, I think this kind of publication and draws quite a lot of people's attention in drug discovery. And although in the paper published after they make some statistics, they don't really conclude this is a direct consequence or direct results when people are paying more attention on safety pharmacology and they have less and less side effects. But according to the results and the statistics data, indeed they find some after 5 years later, they find some lower attrition rate and safer medicine in the world. So of course this is thanks to everybody's efforts and contribution. But such kind of arousal or awareness of safety and of targets, I believe that such kind of information and knowledge also contribute quite a lot to reduce the attrition rate and creates safer medicines to human beings. And here we can see, according to the 2024 paper, concluded from 18 major pharma and biotechs, they have a similar practice. How large is panel size should be considered? How many targets should be selected? So we can see when we are in the very early stage, like lead generation, usually they use less panel size, meaning they select less targets to be screened. But with the workflow and progress, like lead optimization, they will have a more larger panel size and more targets selected. And the final candidate selection, we can see the target selected drastically increase. And the trend is similar like the regular CR provide, like in Eurofins, we provide the safety 44, 87, and spectrum screen. So the panel size and numbers are very close to what we are providing. And this is already kind of industrial practice when we are working in different stage. If we are in early stage, we have more compounds, then we'll do less targets, but I screen more compounds. But when I'm very close to IND, very close to the drug development stage, going to the animal, and also even sometimes coming back from the clinical part, and maybe I have a less compound, or I want to rank my compound, the candidate from rank 1, 2, 3, and then I will do kind of a large panel size to find out the best one for the IND and go into the clinical trial. And here I just like to take a few examples, like industries or academy research institute, they are using such kind of a panel practice. So we can see there are some examples, some this is kind of antidepressant compounds. So they use the safety 44 panel and with some additional targets to do their safety screening. And they have tried using such Eurofins discovery service to optimize their compound. And also for the larger panel, same. So this is for novel sigma-1 receptor antagonist for pain management. Also, they use Eurofins safety 87 for their safety assessments and try to optimize their compound and find some result and make their interpretation of their compounds. And they say their compound 10 have a minimum binding to human hERG, and this is highly related to QT interval prolongation. So such kind of safety panel with proper interpretation, that will be good for the create better compounds. Yeah, and this is another one they are using a large panel. So this is another research institute. They are working in the Huntington disease. So they are making their compound from compound 2 to compound 27. So they use a Eurofins spectrum screening panel and the ADR enzyme panel. Totally, they use they test more than 240 different CNS-related targets and to optimize their compounds from 25 hits to 12 hits. And they claim that will make less off-target liability and make their compound become safer. And in the end, another example is actually from our internal case study. So we took the well-known and already widely used approved drugs. Very old drug is approved in 1976. It's a loperamide, or we call this Imodium for anti-diarrhea. So this is opioid receptor agonist. And in 2016, actually, they got some formal alert regarding kind of serious heart problem associated with overuse or abuse of loperamide. Because we know this is opioid receptor agonist. And under regular situation, they don't have a very strong blood-brain barrier penetration. But when we have abuse case, and some of them, they can penetrate to BBB and cause some kind of opioid-like effects. But meanwhile, they will cause some kind of heart problems and associated with such kind of abuse. So we use this compound trying our safety panel 87, and we do find quite of a lot of targets. And you can see they have some hits on the CAV 1.2, some hits on HERC, on CAV 1.5, and of course like a dopamine receptor, alpha receptor, and of course their on-target opioid receptors. And to the modern medicine, maybe in 21st century, such kind of compound maybe will be difficult to pass the IND and to be approved. But this was like 50 years ago, so we were not really aware about such kind of target effects. But nowadays we have a more and more awareness and better technology to detect such kind of target effects. So according to our internal case study, we can find such kind of off-targets, including alpha receptor, CAV 1.2, beta receptor, serotonin 2A, 2B, HERG, and sodium channels. They are all related to cardiovascular risk, such like blood pressure control, bradycardia, tachycardia, heart failure, vascular thrombosis, and some cardioarrhythmia. So that could be highly clearly explain why FDA or clinical observation we find such kind of heart problem according to such kind of abuse due to their off-targets all related to cardiovascular effects. So this is a proof saying that such kind of off-target screening part could be really predicts or exclude the potential safety risk in the later phase, or even to explain the clinical observations. Okay, so yeah, this is just, again, we review the history and we talk quite a lot about the 2012 paper, both paper in Nature Review Drug Discovery. And now we are going to the last year's paper, the latest one, Brennan's paper in Nature Review Drug Discovery, talking about some kinase dominates safety assessments. So based on the 2012 paper, the 44 targets, in 2024, they make some kind of additional targets, mainly from the kinase. So they say this kind of kinase dominance addition. So they include another 33 molecular targets, and 19 of them, they are kinase. So this is a kinase dominant, but also they put some epigenetics targets, metabolic and inflammatory focus, and from some database from the 18 major pharmas. So another additional 33 novel targets, not novel target, but the targets will be included. And they say these are novel safety pharmacology targets beyond Bauer's paper 44 targets, and it's a kinase dominance. As mentioned before, in the past 10 years or past decades, we have more and more drugs they got approved. Actually, their targets is on kinase. This is also related to we have more and more research and ambition in the oncology field. So many oncology field medicine, their druggable targets on kinase. So that's why in this new safety panel and the new safety suggestion, they include a lot of kinase into the new panel suggestion. Okay, and yeah, I just like to mention, actually, if we are working or if you are studying kinase, you will know, usually the kinase inhibitors, they will bind to the ATP binding domain to try to affect or to block the ATP binding, and in the end, block the activity of kinase. And such kind of ATP binding pockets, they are quite of like very similar across all the kinase. So they have a kind of very similarity in structure or in their binding pockets. So that's why there will be kind of a lot of crosstalk if we are working on the small molecule, focus on kinase. There will be a lot of crosstalk if you are targeting on kinase A, but probably you have some crossover to the kinase B, C, D, E, F. So such kind of a crossover is very often when we are designing the small molecule onto kinase. So that's why they think kinase, as long as we have a more and more kinase-related kinase inhibitors, so they think this kind of effect on the kinase should be paid more attention. So that's why they have new suggestions with kinase-dominant safety panel target suggestions. Yeah, so just in the previous slide, we talked about third generation about safety. Still, we follow the workflow from hit finding, hit to leads, lead optimization, and candidate selection. We have a tier strategy from tier 1, tier 2, and tier 3. And with less targets from 18 core panel, when we just finish completing your high throughput screening, you have more than 100 compounds. You want to do a very, very quick but very critical screening, and you can go for the 18 core panel. But also you can start from if you have a hit to leads and maybe the middle part of a workflow, and you want to just know the very classical 44 targets, and you could select safety screen 44 panel for the mostly binding and enzymatic assay, or you can choose the safety scan 47 panel, mostly for the functional evaluation. And after you have leads, you optimize your leads. You have a better admin properties, and you think this is very good and I want to test more targets. Still, you can use our conventional 87, or you can, based on the new suggestion, you can, based on the 18 core panel and plus another 59 to make finally a 77. Or you have done the 44 and you add another 33 and make finally the 77 core extended panel. That could be also possible. But it depends on you are working maybe more CNS and cardio. You have more concern on this, maybe you can choose 87. But if your compound is related to kinase, maybe you can choose the new 77. Such kind of a selection. We have all different options for your selection, so you can think about what is the best for your safety assessment. And in the end, when you are going to the candidate selection, that will be the final tier 3. And Eurofins Discovery, we have a bioprint panel, it's around 120. And we have a spectrum screen panel, it's around 160 targets. And you can do a very detailed and complete screening for your final candidates. Okay, and there are just some example and comparison regarding the classical 44, 87, and the new suggestion 18, 59, and 77. And if you like such kind of information, you can contact to Chun-Yao, and they will provide such kind of information to you. Yeah, and just one slide I want to mention about the new paper from 2024, and talking about kinase. And in our experience, we talk with a few clients. They really pay attention, and they have their concern about the safety. And also, many of them, they are working in kinase. So some people, they say, you are adding 19 extra additional kinase target. This is very good. But I'm already a kinase guy. I'm working quite a lot in kinase. So do you have other solution? Because I'm not satisfied with only 19 or 20 kinase, but I want more. So if you are really working on kinase, in Eurofins Discovery, we have a KeynoteScan or a kinase profiler. They could be binding assay or activity assay for your selection. you can, according to your requirements, you can pick out either of them. And they have a more than 400. So for the KinoScan, we have a more than 480. And kinase profiler, we have more than 430. So it's really like a very wide and broad coverage of all the kinases. So if you are really working on kinase inhibitor and your small molecule, your compound is focused on kinase, maybe you can start from here, the kinase profiler or the KinoScan, and then you have a more and more detailed information in your kinase part. And later you don't need to worry about the safety concern on the on the new panel, adding on another 19 kinase inside. So that could be another alternative. And if you are really a kinase guy, you can think about and consider our kinase services in Eurofins. Okay, so once we finish our secondary pharmacology data, and this is very useful for the regulatory review. So when we are have our final report and our interpretation, there will be some experts we need to describe, such as the target panel. So how many targets, what target we select for our off-target evaluation, maybe some G protein transporters, enzymes, new hormone receptors, etc. And how many targets we test. And such kind of target, if I have some off-target hit, how can I interpret? Or will they be very critical, dangerous? Or this doesn't matter, so we need to do our own interpretation. And also we need to describe our methodology, like this is a binding assay or functional assay. Both binding assay, functional assay, they will have their pros and cons. And usually we encourage clients just do binding and at the beginning and do the functional follow-up later, and then you don't miss any information. And on the methodology part, you have a more details. And then once you have the result, okay, I have a good hit on my primary on targets. Also, I have some off targets, but then we have to think about off targets. Do they have enough safety margin? Do they have a large difference, huge difference of their IC50 or EC50 or binding affinities with the on-target part? And what is the margin? And also, do they have a good tissue penetration? For example, they don't across PBB, and that would be another thing. Or do they have high protein, plasma protein binding? Do they have enough stability in different kind of physiological environments? And will such kind of situation affect the final off-targets effects? And then we need to do overall data interpretation from the primary on targets, secondary off targets, and considering also about the tissue distribution, admin properties, and the application of different conditions and disease status. And then we can do our explanation. And then, of course, in the end, we have a discussion section and talking about biological significance of our binding, how potency of the off-targets, and relative in vitro potency activity versus our on-targets. And also the IC50 Ki value, that's also very important index. And then correlation, the in vitro finding and other in vivo or even clinical findings. And then we do our whole overall discussion. Yeah, and there are just some objective of secondary pharmacodynamics, just take-home message for your information. So we identify such kind of effects, and we can predict potential adverse effects and provide enough information for the treatments of adverse effects if already we know the targets and the MOA. And then we can detect some functional changes or toxicology studies. And then also it's better to understand the side effects, adverse effect, and MOA, and when during the clinical studies. But also, honestly, I have to say, in the very common biotech practice, a lot of clients actually they don't do this in the very early phase. But when they have an animal study, they see some adverse effects, they have a diarrhea, they have some cardio problem, or they have some strange or abnormal behaviors, and they come back to me. I have such kind of observation. Can your safety panel and your safety service still available or still useful? if I just go to animal study immediately and I want to come back to the in vitro part? So the answer is yes. When you are observing some like side effects or some abnormal behavior from animals, still you can come back using the safety screening panel and see what is your off-targets. And according to the off-targets report, and then you can try to explain or link to the adverse effect you find from the animal studies. That could be also a kind of practice if we are going to the animal study immediately, but we want to come back to the in vitro study. Then this is also possible. And also there are some conditions that could be such kind of safety study probably will be not really necessary if our medicine is from topical use and with a very minimum systemic absorption. So maybe you just use on the skin or the very topical one. You don't have a very like concentration inside your body. Maybe you don't need. And also for the end-stage cancer patient treatment, for some like a cytotoxic agent, already we know this is very toxic, but for end-stage still we, considering about the risk and benefit, maybe we still need to go to treat the cancer first, and safety maybe that will be another issue later. Or we have some biotechnology-derived products, like we have a very high receptor-specific targeting, like we have antibody or some new modality with very high tissue or receptor affinity, then we don't need to do such kind of safety assessments. Or this is just a new salt form with comparable PK or PD properties, and then we don't need to do such kind of safety anymore. So there are also some conditions could be excluded from the safety pharmacology studies, just for your information. And for the long run, we have, this is kind of conclusion for the general safety. So for the in vitro safety pharmacology, of course, we like to do this as early as possible and as wide as possible. And also for the in vitro screening, first is comprised of 3R word chain. And also, as mentioned, we do the binding assay, and after we do the functional follow-ups, that could provide more translation to the in vivo studies and clinical trials. And the most important is how we collect the data and how we explain the data. And that will be very important and really rely on all scientists and working on the drug discovery process. And I think we still have a few, maybe another 10 minutes. I will go quickly through the CNS safety and some cardio and carcinogenicity risk assessments. And among the safety assessments, CNS part is also very difficult part to handle. So from the pie chart, we can see from clinical safety study, we have only 7% they are related to CNS. But when we go to the clinical part, actually there are 34% side effects related to CNS. So you can see from preclinical to clinical, there are 5 times difference. So why there's a huge difference? I don't see any problem in the preclinical part, but I see a lot of CNS problem in clinical observation. And I want to come back to see what happened. Actually, for the CNS adverse effect, there are a few limitations or some difficulties we need to manage. So some kind of adverse effects, such like headache or dizziness, we cannot easily find from the animal study. The animal will not tell you, okay, I got headache, I feel very dizzy, or something like cognition or auditory damage is also very difficult to be found in animal study. Maybe it's kind of chronic and long-term damage, so we cannot easily evaluate. So that makes some gap between the in vitro study, animal study, and clinical study, and especially some effects you cannot observe in animal. But when we are going to clinical trial, and patient they will complain. Patient will say, okay, I really got headache, I got sedative effects, or even they have a drug abuse liabilities. So this such kind of information animal study cannot tell. So that's why we need to move earlier to the in vitro part and try to find the related off targets and to explain or to manage the clinical observations. So if we can see the right figure, we can see there are some like the ranking of the CNS adverse effects labeled in the FDA database. So the number 1 is suicidal ideation, and number 2 is sedation. Number 3 is abuse liability, and number 4 is seizure. So like seizure, of course, we can see some animal behavior, and we can observe some seizure from animals. And abuse liability is a little bit difficult, but still we have some animal models we can try to see if they have a abuse potential or liability. And sedation, maybe you can see from the animal, there are some kind of maybe the motor activity get decreased, but sometimes it's not very easy to be seen. But for the number 1 suicidal ideation, actually animal will not tell you, I feel very depressed today, I want to kill myself. So this is not easy to be detected in the animal study. And that could be the very difficult part to manage. So from such kind of perspective, of course, we try to provide some kind of in vitro off-target assessments to better predict, or even later we see from the animal or clinical study, we can explain what's happening. So yeah, let's jump to the drug abuse. Actually, for the drug abuse, it's kind of not only for the healthy problem, but also for the social problem. So this is already listing to the guidance. So in the FDA guidance, there are kind of clear guidance regarding how we assess drug abuse potential for this compound, from the chemical structure to the binding to certain critical receptors such like dopamine system, serotonin system, opioid receptors, cannabinoid receptors, NMDA receptors, or some major transporters. There could be, if you have such kind of MOA or binding, and then you have to think about maybe you have a potential for drug abuse. Of course, later you have to do some functional assay to find out if they are agonist or antagonist, and what is their MOA, and then decide if you have a really potential for drug abuse. And later you go to the animal study for PK, for some CNS-related behavior model, like reward effects, like physical dependence. And then later you have a clinical trial to healthy human and see if you have the trends of drug abuse. So there's a very detailed guidance regarding the drug abuse potential. And what we can do here for the in vitro part is from the chemistry to binding and to receptor functions. This part we can easily, or we can just use some in vitro methodology to find out the binding and function of the potential receptors or ion channels that could be related to drug abuse. So similarly, for the seizure liability, this is also kind of a very often CNS side effects. So not only for the neurological compounds, for any compound that can penetrate to BBB, they have such kind of situation. And clinically, we find like 6 to 9% seizure cases are due to the drug toxicity. So if you in the hospital you see like 100 seizure patients, maybe 5 to 10 of them they because of the drug toxicity cause their seizure, but not from their like original healthy condition. So that could be also another issue. Of course, for the seizure liability assessment, we have molecular targets. Also, we can do later like electrophysiology on brain slice or zebrafish. And also in the rodents, we can do EEG and behavior observation to assess the final risk. So for the in vitro part, we can provide a pharmacology study like in vitro binding assay, in vitro functional assay, and to provide some information regarding the receptors they will be involved into the seizure liability. And if we are really focused on CNS, and as mentioned, we have number 1 suicidal ideation, number 2 sedation, number 3 drug abuse, and number 4 seizure. So if we are really worried about the CNS side effects, if I know my compound is for neurological disease, my compound is have a high penetration in BBB, high distribution in brain, I really worry about the CNS. So we can provide such kind of CNS target panel to coverage most of the top 4 CNS adverse effect receptors or channels for your selection. So yeah, this is a target list. You can make information, or you want more information, you can contact Chun-Yao for the detailed assay list. So such kind of molecular target selection mostly cover the top 4 CNS side effects. So that could be an option if you really want to study in CNS. Yeah, and then this is the conclusion of the summary for the CNS safety assessments. So during the hit finding, of course, we want to know the MOA and we want to do find out our on targets. We have a different kind of CNS related or therapeutic area related panel like PAN, AD and PD or neurodegeneration. But when we have the leads or even the preclinical candidate, we want to assess their CNS safety concerns. We have a CNS target panel, drug abuse panel, or residual liability panel, and to provide a better assessment of safety in CNS. And one or two slides I just mentioned, I highlight a little bit more regarding the cardiovascular safety liabilities. So we know HERG is very important, but except HERG, still we know the CAV, sodium channel and calcium channel, they are also very important in the action potential in cardiomyocyte. So not only the HERG channel, but the sodium and calcium channel, they will be involved. So nowadays we have a more different kind of technology, including a lot of electrophysiology or MEA, cardiomyocyte, and the other phenotypic assays. It will be easier, help us to easy assess the risk of pro-arrhythmic effects from not only potassium channel, but also sodium and calcium channel. And also in Eurofins Discovery, we have a biomap, such kind of phenotypic platform for not only the cardio part, but also the vascular one, like thrombosis, like a vascular toxicity. We have a different kind of phenotypic platform to help our client to assess cardio and cardiovascular safety assessment. And here you can see some example regarding the sodium channel, calcium channel, and the potassium channel. We have a different kind of electrophysiological platform to provide the evaluation of such kind of channels, seeing if your compound they have some interaction or some functional effects on the critical channels that could be alter the cardiomyocyte's action potential, probably with arrhythmia or from some abnormal contractions. By such kind of platform, you can easily assess your cardio safety. And also from the target base for the heart rate, blood pressure, cardiac contractility, and also structural cardiotox, they are all different kind of molecular target being involved. So if you are really working for the cardiovascular safety, you can also think about or consult us for more target selection for your cardiovascular research. And another part for carcinogenicity, but we have another webinar a month ago talking the details about carcinogenicity and why this is so important and how it's related to the in vivo study and also to the to the reduction of animal studies. So I will not go through all the details, but if you are really interested in the cardiogenicity study and what is the in vitro alternative way, you can refer our previous webinar focus on the carcinogenicity risk assessments. And just like to show, we have a case study when we select such kind of carcinogenic molecular targets. We use different kind of carcinogens to evaluate and to confirm our ideas and target selection, they are correct. And this target selection has been already cited by a few papers, and we are talking about quite a lot in the selection of carcinogenicity risk assessments and how we can make this better. So Eurofins still we are improving our target selection and try to provide a better solution for carcinogenicity assessment for our clients. Yeah, so finally, just like to mention the in vitro safety screening methodology is very important nowadays, especially we have FDA modernization from 2022 the 2.0 to 2024 3.0, and last month we got the FDA announced to plan to phase out animal testing requirement for monoclonal antibodies and other drugs. So reduction of animal usage comprised to 3R and use more humanized models, humanized in vivo models, or some alternative technologies such like in silico prediction models or iPSC organ on chip, such kind of technology. will be more and more important. And we try to reduce the use of animal and try to fill the gap, as mentioned, like some CNS side effect. We cannot easily see this from the animal study. Then we need such kind of new technology. And the in vitro safety screening, in vitro platform, and also the humanized models will be more and more important in the future drug discovery and also for the safety assessments. And yeah, there's just kind of a conclusion for today's seminar. So we are working quite a lot in safety pharmacology and try to make a safer medicine for human being. So for such kind of safety pharmacology study, we can eliminate the potential liability in the early phase of discovery and reduce the risk in human and lead candidates attrition in the later clinical phase. And also such kind of information can directly help the chemistry in their lead optimization. So when we optimize the compounds, we can eliminate such kind of targets and making the compound safer. And also such kind of information can help us to rank our candidates. So which are rank 1 for the clinical trial and which rank 2 or rank 3. We can do our own ranking due to the information. And then we can mitigate the risk associated with the selected candidates and also make our own internal decision making for our workflow and decide if we go to the next stage or not. So such kind of information is very important for drug discovery. And yeah, in the end, this is just the same page. You can, if you want more information about what kind of panel service Eurofins can provide, you can contact Chun-Yao for further information. And yeah, this is another example for our different kind of screening panels from general safety, CNS safety, and carcinogenicity safety. We have all different kind of service for your option. So thank you for the listening. I'm hoping we'll have a good and better medicine for human being. Thank you so much.